Should we start?
Yeah.
Okay.
It gives me great pleasure to introduce Anna again, who's going to be talking to you about her experience, partly, presumably, about academia and the partnership with pharma. So, you've been involved with the Marshall Centre originally, and then it's spin-off Biologica, and then...
On deck.
Biologica was
yeah.
Tikawa, On Deck, and then more recently you branched out into your own...
Well, Biotome as a startup and a vaccine program at Telephone Kids, which I talked about last time.
Because it's such a hazardous environment.
It's hard work. Startups.
You've got to do everything, haven't you?
You do got to do everything. Yeah.
All right.
Thanks, Chris.
I'll see you later.
All right. Dim these lights. All right. It's only three of you. We should just go and have coffee. Okay, so today the lecture is on roles of academia and pharma in partnership. So I'll talk a little bit about the current project that I run at Telephone Kids, which I when in my first lecture we touched on strep A vaccine program, and that's a really good example of academia or academic environments and pharma partnering to advance a vaccine that may not otherwise be advanced. But it's not in my slide deck, so I'm going to today talk you through about some of the reasons why this works and how there's been a shift in sort of the field over the past probably 10, 15 years. In the beginning it was viewed very separately, you were either academic or you were pharma, and now I think the pressures of R&D, the costs actually make it a really nice partnership because the different entities bring different things to the table. So before we start, I'd like to acknowledge that we are on the Whadjuk Noongar Budja land, and I'd like to pay my respects to elders past, present and emerging, and thank them for looking after this country that we work, live and play on. So today, the slide deck's a bit intense, and I've made that intense for you to have, but I'm not going to talk to everything on this slide. Hello. But the key objectives are, what are the different roles that different public institutions, and that encompasses academia, so universities, but also hospitals and environments where government is funding, but there is research and clinical work taking place. So just bear that in mind. We'll talk about benefits and challenges. I'll give you some high-level examples of some drugs that have originated from academia and are now blockbusters and, earn millions and millions of dollars for pharma. And then I wanted to put forward the case study of Moderna and the COVID-19 vaccine, because it's a really good example of the mRNA technology actually was developed in academia 20 years ago. It's been a real evolution, and then the US government funded Moderna to develop its vaccine as well. So it's a really nice example of where you marry both. You can go fast and you can actually end up with a successful outcome. So why is pharma interested in academics? So I think in the very first lecture I gave you, the cost of developing a vaccine, for example, is now, I just looked up the figure, it's now in excess of $1.2 billion. So that is just to get through phase 1, 2, 3 and put a product on the market. And it's that really critical path activities, manufacturing included, technical assay development, analytics. What it doesn't do is give you information on disease targets, disease mechanisms, pathogenesis, surveillance of infectious diseases, for example. So for the strep A program that Telephone Kids has partnered with GSK, we're developing a vaccine to prevent strep A pharyngitis with the view that we want to prevent rheumatic heart disease later on. We don't know the contemporary strep A sore throat rates in Australia at the moment. COVID has shifted that. So one of the things that we're undertaking as the academic or the public health partner is that we are doing the surveillance. So doing epidemiology surveillance of children, urban living children in Australia. We've got now 3 sites popping up, Perth, Melbourne and Adelaide, and we're looking at that data. So that's something that a farmer, these are field epidemiology studies, they're not really going to do because it's not on their critical path, but by doing that for them or with them, we can get real-time contemporary data, and that allows us to power clinical trials more effectively. We can design better vaccine trials and make sure that we can actually measure those clinical endpoints. And for this vaccine, it is measuring a culture-proven, serologically proven strep A sore throat as an endpoint. So that's been really interesting. So one of the reasons pharma is interested in academia is because the high cost of R&D. So for them to do R&D in-house is very expensive, time-consuming, and it isn't, they're not focused. I'm just going to grab my pointer, if this works. Pharma is not geared to do basic research, the blue sky stuff to follow those questions on biological targets and really this disease pathogenesis, understanding the disease mechanisms. So I think that in the first instance is a really good partnership. And the other thing now that's happening in pharma is that the cost of R&D is high, so then your pipeline starts to diminish, and unless you can maintain that level of basic research, you start to not have a product pipeline feeding through. So the reliance on academia to actually build product pipelines is strong and large, and there's lots of examples that we'll talk about where this has happened. Pharma are actually focused on the translation. They are drug developers. They want to, get the lead target, get it optimised, go through the development pathway and get it into licensure. And there's many examples of drugs and vaccines licensed where the mode of action is not actually understood. It's because you don't actually need to know it. If you have a clinical effect and you can, prevent disease X with product Y, it's enough for a regulator. It needs to be safe, of course, and all of that. But actually understanding, unravelling how it works is sometimes not as important as having a product that prevents or has a biological effect. So pharma generally rely on academia now to identify and validate novel drug targets, develop the assays, develop the animal models to test this, and very importantly now academia also provides access to new financing models because academics have access to government funding, to grants, to philanthropy that large pharma or even biotech to some extent don't have. And there's this perception. So I think the take-home message here is that partnerships do bridge the innovation gap. It allows you to look at a problem more globally or holistically rather than just focusing on the simple development aspects of pharma programs. But having said this, academics have been collaborating, for decades with pharma. It's complex, and we'll talk about what some of the challenges might be. But as I've mentioned, academic institutes offer a lot. So they offer access to basic research, access to deep knowledge, to explore those really important scientific concepts that you might not have the opportunity to explore if you are a R&D program within a pharma, and particularly this mechanisms of disease. I've now mentioned that several times. It's quite important. Ability to build a pipeline and really identifying new targets. And one of the examples that I'll talk about is Humira, so the anti-TNF alpha monoclonal. And it was a really beautiful partnership, and it came out of academics when they first discovered TNF alpha being involved in inflammation, and then, so I won't jump my slides, but there's an example of that. The other thing, and I think you'll hear from Peter Richmond about this, is that academic institutes and hospitals offer access to clinical trial, to patient cohorts, patient samples. So there is this ability to actually partner to conduct those phase 1, 2, 3 trials, or at least components of it. Academics also offer peer review, rigorous peer review of the research, and we've already talked about access to funding and trial participants. And the main difference between academia and pharma is that pharma and biotech are very focused. They're focused on drug discovery, drug development, manufacturing and regulatory. So really funneling onto that critical path. But what we can do by collaborating is leverage actually all of the academic strengths and all of the deep focus on drug development that a pharma brings so that we can accelerate. So we need to be able to move quickly and have the best use of our funds. So this, I pulled this out, and it's just an example. It only goes to 2015, and I didn't have the figure. I couldn't find the figure for 2015 to 2022. But basically, you can see here that, you know, there is this, so number of drugs approved over time, and this is the cost. So, you know, 58 billion was spent on R&D in 2015. Where are we? And this is increasing. So, but what has happened since 2015 is that although expenditure has increased, number of drugs approved has decreased. So, and that is really a reflection of the really increasing costs of R&D programs, really long timelines, and despite more spend, there's less drugs approved. So it's becoming harder and harder, and the market is more competitive as well. So I wanted to put up this, and this is the US National Institutes of Health, or the NIH, has a really nice model, and they've actually been involved in many translational research programs where their discoveries have ended up with drugs on the market licensed. So, and the reference is here. So it's an interesting paper. It's a little bit dated, but the concept is the same. So what the NIH does is that they have this four-pronged model. So that they have pharma, obviously, they have academia, they have NIH, which is the government body providing funding, and then they leverage that and have philanthropy as well. So, and by doing this, by sort of putting the four pieces of this jigsaw puzzle together, you have a four-way partnership, and what that does is actually accelerate. So you can overcome the challenges that drug discovery has by bringing in money, which is not only government funded. So, you know, moving away from that perception of conflict of interest. Are you being paid by pharma to do what research that they want you to do? You have the academic here who is able to pursue, you know, the blue sky stuff, the conceptual stuff, and try and find new targets. Obviously, the relationship has to be that pharma will come into this picture if the academic is doing work that is of interest to them. So, is it a disease area they're interested in? Is it a signalling pathway, or is it a particular molecule that they've identified? And philanthropy is a really nice way of accessing funds unencumbered. And so the Strep A program that I lead now at Telethon Kids is actually exactly this. Instead of the NIH, we have the Australian federal government putting in $35 million through an MRFF grant, and we've actually leveraged and brought on 2 philanthropic donors on the back of the government funding to the tune of about 15 US million, and that enables us to actually build sort of a funding consortium that doesn't look like the government is funding a pharma program, but there is a whole network of people that have acknowledged that it's a problem that is worth solving. So that's it's quite nice. So some of the examples of drugs, I've just listed 3. There's tons of them, and there's a couple of tables in the slide deck which I'll leave with you. But you know, all these 3, these are all small molecule drugs. Pregabalin is a pain, so it's an anticonvulsant, it's an anti-pain, so it's an analgesic, and an anti-anxiety medication. So it's you know, the wonder drug. It actually came out of Silverman's lab at Northwestern University. It now is, worth, it's a blockbuster drug, so over $1 billion a year market. Mtreva, same sort of thing. This is it's a reverse transcriptase inhibitor, so it's used as an add-on therapy to treat HIV infections. Came out of the Liotta lab at Emory, and Pemextred is it's a chemotherapy, also known as Alimta, came out of Princeton University. So just all small molecules, and these guys were obviously, pregabalin, the Silverman lab was looking at pain and how to prevent chronic pain. Liotta lab was looking at HIV, and the Taylor lab was a cancer and immuno-oncology lab. So these discoveries were made in the universities and licensed out to the respective pharma that now have them. And this is just a list, again, out of the open source journal, but certainly not comprehensive, but it gives you the example of, and this is very US based, so I have to say the Australian, we bat above our sort of weight for the size of our population, but we certainly don't have as many of these deals. But you can see here, Altima, Princeton, Eli Lilly. So you can just see all of the universities here and then all of the large pharma that have come on board at various times. In my experience nowadays, pharma generally like to have clinical data to come to license something in, unless it is really, if it hits the target. If your preclinical data is so strong that it's black and white, hits the target, and immuno-oncology is really now a hot field where if you can find a druggable target and chemically synthesize the component or the compound that hits that target, this would probably be licensed very quickly based on in vitro model data even. So and these are just a few more examples. You can see private sector, public sector funding. So GSK and Harvard, they put in 25 million over 5 years for heart disease and cancer. So these are examples of. So the slide before was actually products that have emerged out of partnerships. This is an examples of how partnerships can develop, and the funding that's been allocated to these things to be able to actually just have the next field of discoveries or the next line of drugs. So you can see sometimes it's actually significant. So immuno-inflammatory drugs, these are quite topical, and 25 million over 3 years. So and that's a combination of Harvard University attracting funding through their academics, and GSK coming on board and providing probably cash injections, but also all that in-kind support, because what pharma does bring is access to a suite of tools that may not necessarily be available at institutes, such as analytical assays and for a vaccine, for example, simply providing you the vaccine to be able to test in your models. So have a look at that. There's a few examples there. And this slide is again not too recent. And here what it denotes is public sector research institutes. And as I mentioned, this includes not only universities but small MRIs and also hospital-based centres. But you can see here that the number of new entities that have received a priority review at the FDA or a standard review, and these are the all approved. So 483 drugs that have been approved in some form at the FDA. So very US based. So it's again another example where you've got huge amount of approvals, and more than 10% of those have come out of the academic sector, which is really, really high. And interestingly, this priority review, so 20% of the 209 FDA approvals for a new molecular entity came out of a public sector research institute. And what this priority review means is that you can apply to the FDA for a priority review, which means that they will review your package 6 months ahead of time. They guarantee this 6-month thing, and that means that your development pathway becomes 6 months shorter, and if it's licensed, you're on the market 6 months faster. And that 6 months in sales is enormous. So, these priority review vouchers are worth, the last one and it was an Australian company called Medicines for Global Health out of Melbourne, run by a guy called Mark Sullivan, a really clever guy. He got one of these for his discovery, he's built a model to treat river blindness in low middle income countries. So he was able to in-license a treatment, ivermectin I believe it was called, and managed to get actually a priority review voucher, which he then pocketed and then sold on for $100 million US to another farmer that wanted this 6-month acceleration pathway. So my point is that it's really valuable to pharma to save 6 months time. So the next couple of slides are just tables, and I'm not going to talk to them today, but go through them. It's mainly for interest. So these are 444 drug discoveries by country, and I just want to note, even Australia, we do rank. So we've had 14 drug discoveries lead to licensure in the world. Of course, this is much lower than the US. I mean, the US ranks at 293 of those 444. So the US is the place that drugs get licensed the most, but just goes to show that, discoveries can come from anywhere. Israel also is pretty high, and that's because their governments and their organisation, their country is geared to actually promote this development and really try to accelerate medtech and product development. So a lot of it does depend on how the strategy and the view of your government. Oops, wrong way. Again, this also just for your interest, these are the top institutes that have performed the best or have had the most discoveries turn into products, and no surprise the NIH is right at the top. These are all US-based mostly institutes. I mean, you've got Canada there, you've got Israel, and individuals, I guess that means just non-individual people that have not necessarily been affiliated to any particular research institute. So the challenges and the benefits. So I think the benefits, there's many, and I've tried to just keep this to 5 each. There's more rapid identification of relevant biological targets, and relevant is really important. We can find something happens in an animal model. We academics work in the preclinical space a lot, but unless you can relate it, your increase in product X, unless you can relate it to prevention of a disease as a clinical endpoint, the question becomes, is it biologically relevant? And I think understanding these disease mechanisms, and in particular for vaccines, host pathogen interactions, and looking at the genome of the host and the pathogen, looking at the structural biology of the proteins that are expressed on the pathogen, all of these things help us to better understand how does the pathogen cause the disease, or how does the molecule, the target, how can you drug the target to prevent the disease? That all helps go towards this identification of relevant biological targets. So really important. I think this is particularly important as well. Researchers get critiqued, really peer-reviewed and published, and this altogether de-risks projects. So the more review of a particular result, or say you found a new molecule, the more review that has had, the more validation that you get that this is in fact real. Other labs will try and reproduce your data. So that I think is all good. And this diversification of funding, so diversification of funding in that it comes from governments, it comes from philanthropy, it can even come from high net worth individuals, so individuals that, are wealthy that want to contribute back. And I think, I haven't put it on this slide, but now there's the whole ESG, so really trying to work towards social impact and not only profits for companies. I think, the measurement of success has changed from, can you offer a 10x return on investment to how do you impact where perhaps such large revenues are not involved. The challenge is now it's complex, it's really complex. There are different kinds of pressures on academics versus pharma, and pharma experience commercial pressures. They're very driven by tight deadlines and timelines and trying to get to the market first and fast. So I think if you're first, you've captured the market simply by being first, and then you've also got IP that is ticking, so you want to be able to get to the market and then have as much IP protection of your product before generics come into play, which is usually after about 20 years. And pharma have very clever strategies of trying to extend patent life cycles and add on things so that they get longer protection. The other difference is differing priorities. So for an academic, who you want to publish, you want to have your track record, you want to get that next promotion. So I think the pressures are different in academia versus pharma, because pharma want to keep things closed. They don't necessarily want to disclose what their drug target is or what they're working on. Sometimes they might not even file that patent for several years. So this is where that trade-off with working in these types of partnerships becomes difficult. So this publication data sharing, you really need to come to an agreement before you enter into any partnership on what that is going to look like, so that no party is feeling less or unable to pursue their own interests. And another challenge is also that academic practices and industry standards are quite different. So the level of rigour and documentation that an industry partner might need versus an academic partner are sometimes chalk and cheese. So trying to explain to each side why that level of documentation is required or that level of rigour, so that both parties understand what they need to deliver. And I think nowadays this conflict of interest is particularly relevant because we have such tight subject matter experts and everyone works with everyone. So really ensuring that you're not conflicted, and if you are, that you've disclosed that up front so these partnerships can work. But I do think there's lots of evidence that you can overcome the challenges so that you reap the benefits. This next slide is just the types of partnerships, and I think in the more traditional sense, the first two are how pharma-academic partnerships have been perceived sort of to date. But more recently, we're moving to these collaborative research models, and the caveat there is that you must overcome the challenges that we've talked about before, the data sharing, the publication, the IP, to be able to work collaboratively. So, you might have a PI-led research grant, and that's quite a common approach, where in fact sometimes pharma even put forward the funding for these research grants. And you'll hear from Peter maybe that Merck has a really big research funding program where academics can apply for funding to address some of the issues that they're interested in. So this really supports an investigator's research without any encumbrances or restrictions. It's led by the PI owns the idea, the PI develops the research plan, and a farmer, is listed on the grant to be able to provide, sometimes financial input, but sometimes in-kind support in terms of technical expertise or something like that. But really in the first type of partnership, the PI the academic is in charge. The second type of partnership is really a contract research agreement, and it's a fee for service, and it's really used to define a specific problem. It's usually associated with much, much higher costs because the academic doing this fee for service work doesn't get any IP, doesn't get any, you can agree on some publication date or possibility, but it really is at the discretion of the payer, and the payer is the pharma company here. And for example, like in my career, we've used contract research a lot as an academic, but also as sitting on the side of pharma. And you might, for example, want to test that your product works in an animal model. The example that I'll use is a real example. So we developed a allergy immunotherapy, and this was when I was working for OnDeck with Barry Marshall. So we had developed a bacterial-based immunotherapy, oral immunotherapy, that would prevent eczema and paediatric allergy. But we thought it was a broader anti-inflammatory product. So we wanted to see whether we could actually stop Crohn's disease, inflammation in the gut. And so we didn't really want to go straight to humans, so we then found a contract research organisation in France, and we contracted them to do those 2 animal models of Crohn's disease for us, and we paid, the premium price for that, rather than setting that model up in-house in an academic lab and trying to do it that way. Now, I think there's a lot to be said for contract research, because in my mind, I think as leading that program, I want to minimise my financial waste. So I want to go to a group that has done this model, the model's validated, I know they can do it. Otherwise it's going to take me 3 times as long and probably a lot of grey hairs to try and set that model up in the local animal house, and then will it work, will it not work, why hasn't it worked? So really going to the experts to do what, they do what they do, that's good. But it does cost a lot because, as I said, the other side is not getting anything out of it except for the money that you pay them. So usually the difference, for example, between a contract research fee for service and the same grant the same project as a grant is 3-fold. So, you might put in an NHMRC grant up here for, I don't know, $100,000 or $200,000, and you go for a contract research place and it will cost you 3 times as much. But that's why, it's because that's yours and they don't get anything. The third one is the most interesting, and I think this is where we're going to. We're going to this collaborative research models. It's co-development. It's going through those parts of your project or your project activities together. It's sharing the risk, which means sharing the cost and sharing the risk of failure. So very high risk of failure in drug or vaccine development, and you're prepared to do that. I think we're getting a little bit better now in starting to actually document and report failed studies and failed clinical trials, because that's actually where the knowledge is. If you can understand why something hasn't worked, you might actually be able to find the reason why it could work. And you need collaborative research agreements with options. So, but it is difficult because, for example, who owns the IP in a collaborative research model? a pharmaceutical company needs to own the IP if they're going to invest, millions and millions of dollars later to develop it. So some of the things that we see now is that you enter into these collaborative research models, the IP remains, any discovery IP can, pharma has first right to license, that sort of stuff. And there's some term and condition or some clause to say that, well, if it is successful, if it does end up on the market and it is a product making sales, well then the original academic partnering institute will get a small royalty or something like that. So there are ways to reward this really early investment or partnership. And I have to say, this co-development and risk sharing, the earlier you are in a program, the greater your risk. So the value of your dollar, for example, in a phase 1 or in a phase 2 vaccine trial where you have proof of concept is tenfold more than the value of that dollar when you're in phase 3, when you've already got evidence that it does work in humans, it's safe in humans. Having said that, there are many trials that fail at phase 3 as well because you end up seeing a rare safety signal. For example, the AstraZeneca COVID vaccine, that they didn't see that in the early trials, and then it was this the thrombosis event that actually stopped that vaccine, but that was in phase 3. So I hope that just gives you a little overview, and we'll soldier on. So next couple of slides are again tables of recent industry academic partnerships, and you can see University California, San Francisco with Pfizer, the Burnham Medical Institute with J&J, the German Cancer Centre with Bayer, and Bayer is really focused on cancer therapies. Again, Harvard, MIT with Sanofi. So there's lots of evidence of really successful partnerships, and the one thing that I haven't actually included here is nowadays there's actually these consortiums that are being built. So consortiums of academics, biotechs and pharma where there's a common interest. So it might be developing genetic tools or cancer immunotherapies, but and by a consortium what they do is they come together. So they come together to discuss what are the critical issues in the field, what types of treatments and should they be working on. I won't talk here much because I'd like to get to the case study. So I'll leave this with you. It's just a little bit of extra reading, and the reference is there if you would like to read the whole paper. So the take-home message is together, pharma and academia do have a unique opportunity to create an enduring partnership that will accelerate discovery of innovative medicines. I think nowadays with the extreme costs and all the pressures that come with drug development, you need to actually have both, because having both gives you more information from the pharma and you understand disease mechanisms much, much better. So the current trends are that academia is really easing this drug discovery bottleneck and contributing very largely to unmet medical needs, or to solving unmet medical needs. So now, in the last sort of 5 minutes, I'm going to flip through these two examples. And these are examples of really successful partnerships, but partnerships that have been around, that have taken decades to actually get, Humira, which was the first monoclonal antibody to be licensed, and it is, I think, still to date, the best-selling drug in the world, globally. It's number one. And then a more recent example of the Moderna COVID-19 mRNA vaccine, because the work on the Moderna mRNA vaccine actually started 20 years ago. And it was because of all the background work that they were able to pivot really quickly. So, Humira. The story of Humira first started when they discovered TNF-alpha. And this was done by a bunch of academics in the US, and they developed that the molecule TNF-alpha was discovered, the structure was identified, and it was discovered to be a naturally occurring cytokine and an inflammatory mediator. So very soon after that, people thought, well, if we could block TNF, bingo. You know, if we could block this novel molecule, we have a chance at trying to reverse some of the inflammation that goes on in a variety of diseases, in rheumatoid, in autoimmune diseases, such as Crohn's, such as RA. And in fact, Humira was first licensed for the treatment of rheumatoid arthritis before the label was then expanded to other inflammatory autoimmune conditions. So lots of text for you to read, but the bits that are underlined are, so discovery of TNF-alpha in an academic setting, back 20 years ago now, in the late 90s. And then the next thing that happened was that monoclonal antibody technology became available. So I think this is really important, and this is why you do need academia as well, because you need academia to be working on trying to work out how to do things, how things naturally work in the human body. So when monoclonal antibody technology became available, developing Humira became possible. So it didn't matter that you knew TNF-alpha, we didn't know how to program, how to make hybridomas, how to make hybridoma, B cells to be able to produce buckets of antibodies specific for the TNF-alpha. And then what happened was there was a collaboration between academia and industry, in particular Feldman and Maney in the UK, and they did the real sentinel, the foundational research on the role of TNF-alpha in rheumatoid arthritis. So you can see there was a discovery of a molecule, then we had the technology, and then there was the link between this molecule and a disease. And I think that all took a while, 2 decades to come together. And then once you had that, then of course Abbott put their hand up. Abbott's now called AbbVie, and they acquired the rights to develop and commercialise the drug. So my point here is that if any of these 3 bits hadn't occurred, we probably wouldn't have Humira today, because you needed the link, you needed the molecule, you needed the technology, and then you needed to link the molecule to a disease that you wanted to prevent. And then it took off. So then Abbott licensed in, they established the technology, they created Humira, went through trials, went through regulatory approval. It was first licensed at the FDA and Europe, yes, now for treatments of various diseases, but very first for rheumatoid arthritis. And it was launched for sale in 2002. So again, timeline probably from 1 to 7 was probably 20 years. And then what happened is you expand from rheumatoid arthritis to other indications. So really nice. It's not off patent yet, but very soon it will be. Are there already? Okay, so there you go. Thank you for that. 2 generics. So it is off patent for RA. Is that for RA? Yeah. So that's good. Yeah, you're right. There it is. 2016 it expired, so thank you. I was just 1 slide behind. And this is just the timeline of how it happened. So 1993 is when the technology, so the monoclonal antibody technology partnership started. Still took a while. So I'll leave that with you, and in the last, I know we're over time, but in the last minute, I just wanted to walk you through the case study of Moderna. I'll leave this with you. This is just the evolution of the vaccine platform. Very early on, even in the 1800s, it was live attenuated pathogen. We moved into inactivated pathogen. We moved into protein-based vaccines and subunit vaccines. And then We've moved into viral vectors more recently, and very lastly, nucleic acid vaccines. What we want from a vaccine is we want a B cell response. We also want a T cell response, because T cells help B cells produce antibodies, but we also know that having both gives us balance. So this slide is obviously a COVID slide because you want CD8 T cell activation. For many extracellular pathogens such as bacteria, antibodies, like for meningococcal and pneumo vaccines, they're antibody, mostly antibody mediated B cell vaccines, but you need CD4 T cell help to create, memory, strong, good plasma cells and good memory cells. So really quick summary of the history of mRNA technology. It started a long time ago. It was proposed in the 20th century, and we've made, leaps and bounds, advancements in leaps and bounds. So in the 40s and 50s, the concept of mRNA emerged, and that was when we understood the flow of genetic information from DNA to RNA to protein. Then, in 61, Jacob and Monod, they developed the lacopperon, which gave the evidence that mRNA has a role in protein synthesis. And then there was more work by Nirenberg, Harana and Okia to decipher the genetic code, and this confirmed the role of mRNA in protein synthesis. And then we had more transcription, translation evidence. And then we get to the 2000s, where we have development of in vitro techniques. So again, we knew that mRNA was, that DNA and RNA was around, and we knew that you could go from translation, to a protein, but we didn't have the technologies, the techniques to do it in vitro until the 2000s, where we could then, scientists discovered that you could synthesize mRNA molecules in the laboratory, and then this could pave the way for therapeutic application. In the 2000s to the 2010s, therapies for and firstly, mRNA was used for gene therapy, but it was found to be actually quite toxic, vaccine development, and also cancer immunotherapy. And then we moved to trying to develop vaccines for influenza, for Zika, and importantly for SARS-CoV-1, so for MERS and SARS-CoV-1. So really long history. I'll leave this for you to read, but basically the next thing that needed to happen is, we worked out, or the scientists worked out that you could naturally modify RNA, and it alters or it loses its immunogenic potential. Then they worked out that you could increase the translation in vitro by making some nucleoside modifications, and then you need to package it. So then lipid nanoparticles. So my point here is that you actually need several things to happen before you can have actually promising nucleic acid delivery. It emerged from the NIH, so it was, so mRNA technology was, the basic research was done under Barney Graham's laboratory in the NIH. The government has poured billions, and this is most recently, so this is just the COVID numbers, the funding that was provided earlier on is, I haven't got there, but it would be billions and billions, hundreds of billions. And so Moderna merged out of this partnership between Barney Graham's lab at the NIH and Moderna. And Moderna actually gets its name from modified RNA. So that's where Moderna, there it is. So in 2010, they named the company based on the discovery that if you modify RNA, you can actually deliver proteins in vivo to the host. So two fundamental discoveries were done in the academic sector. And this is, of course, I'm talking about COVID. So the viral protein was designed synthetically by Graham and his colleagues. And then the concept of RNA modification was developed by Weissman and Carrico at the University of Pennsylvania. So this idea of modifying elements of RNA to allow it to slip past the immune system undetected was actually really important, and this modification allowed the delivery system to be developed for vaccines and also for drugs.
I'll leave you with the timeline. And I think the last closing sentence that I'd like to leave you with is that we shouldn't underestimate the value of basic biology research. And Dr. Fauci said this in the middle of the pandemic. I will leave you with those couple of slides on mRNA technology and immunisation strategies. And that is my closing slide. So the partnerships between academia and pharma really provide the opportunity to advance the frontiers of drug discovery research and promote technology innovation. And the take-home message is you need both. You need both to be able to actually make really successful products that treat a variety of unmet needs.
Thanks a lot, guys.
Yeah, okay.
Thank you.
Is that better?
Okay.
I'm impressed you... oh yeah, no, it does work a bit. Just acknowledging that we're on the land of the Whadjuk people, the Noongar Nation, and pay my respects to the elders, past, present and emerging. And so this is an outline of what I thought I would cover today. And as Chris said, if we talk about the future of all the vaccines that are being developed, this would be a very long lecture. But what I thought I would probably just try and reiterate and give some examples as we go along about understanding the critical aspects of successful vaccine development. And so what do you need to know to develop a vaccine? Why are some vaccines not successful and how can we learn from that? And I'll give a good example how that's really informed future vaccine development. Talk a little bit about challenges for existing vaccines that will come up and some examples that I've been involved in. And then challenges and opportunities going forward. So, and I'll have revisited some of the concepts that we discussed last week or a couple of weeks ago. And certainly, understanding whether the disease is important to prevent. And I think Alma probably would have touched on this. If it's very common and it costs a lot of money, then it's a good potential market. And, the figures that you need to have for your potential market of, $1 billion a year or something to actually develop a vaccine to get it all the way through is really important to be able to provide up front. And without that data, it doesn't get past the preclinical studies. Really understanding how the disease occurs in humans and what protects us from disease, and therefore that helps us select the best candidates for a vaccine. And so that's really critical to understand. And I think we clearly need to understand the structure of the microbe, of the virus or the bacteria or the parasite. And now we know more about that ever. We can sequence any virus that comes along. And, I think a great example is SARS-CoV-2. So that was, within a week that the virus was identified, sequenced and importantly released publicly. So that was an Australian who was able to actually put it out that everyone was able to access it. Within a days, people were working on a vaccine for SARS-CoV-2, and actually within a week they had a construct, and within a month they were producing mRNA vaccines that they would then be able to use. So that's how understanding structure and understanding the function, because we already knew coronaviruses needed a spike protein to infect cells, and that successful vaccines were able to neutralise, to bind to that spike protein for MERS or the original SARS vaccine. But what we also, and so that was really important. And so because you knew both those things, you could go straight ahead and start to produce the vaccines. Really understanding how useful is the animal model in predicting what's going to happen when you actually get to humans. And so really, does it replicate the human disease? So a good example is people developing vaccines for pneumococcal disease, for instance. So this is my mouse model. To get them septic, I inject the bacteria into their peritoneum, okay? That's not a great model because that's not how most humans get infected. We need to have it that it starts in the upper respiratory tract and then it progresses either into the lungs or, for us, into the nose. So how do you do that? Well, for otitis media, where we're trying to develop a vaccine, actually we have to give the mice a flu, and then we expose them to the bacteria, which for us we're looking at a non-typable Haemophilus influenzae, and only then will you actually get the bacteria in the ears, and so then we can test our new construct to see if that prevents it. So really understanding, and in fact that's exactly what happens in children. They get the flu, and then whatever bacteria they have in their nose either goes to their lungs or goes to their ears or gets into the bloodstream and causes pneumonia, otitis media or septicemia. So that's understanding our animal models is really important. And again, going back to the SARS-CoV-2 example, the mouse model actually isn't a great model, and so Syrian hamsters was the final model that seemed to best replicate the lung disease that humans got. And for influenza, it seems to be a ferret is a better model than a mouse. So, and for some diseases, we can't infect the mice with, for instance, RSV. You have to use a mouse homologue, so that a virus that's like RSV but can infect the mouse. So understanding these things is really important. And then this is what you've been talking about with the phase 1-2. What's the optimal vaccine dose, formulation and schedule once you get into humans? Is it safe? And can we get good immunogenicity without getting too much reactogenicity? And so that's really, particularly when we start to talk about adjuvants, so things that we use to enhance influenza responses. We can use MF59, the oil and water adjuvants, or AS01 that we've used for pandemic vaccines, but they do cause some increase in reactogenicity compared to, say, the standard flu vaccine. Or you might use a whole cell or a vesicle, outer membrane vesicle, and that's been used, for instance, in meningococcal B vaccines as a way of not only providing some response to the outer membrane vesicles themselves, but also packaging in other proteins that you can then use for a vaccine. So, but unfortunately in young children, in particular infants, that can cause very high fevers. So as part of that vaccine development, they had to give prophylactic paracetamol to enable the babies not to get too high temperature. And importantly also, when you're doing these early studies, can you get your predictive biomarkers? Can you de-risk the phase 3 study that's so expensive that we talked about? And then finally, you've got the vaccine, it's registered. You need to have enough information to get licensure, but also if you want to have it being used in national or region programs, you've got to convince the public health officials, the PBAC, the Pharmaceutical Benefit Advisory Committee, and the politicians that actually this is a disease that will work and prevent disease in Australian children, adults, and we've had some really good examples where state-based programs for maternal pertussis vaccination and for meningococcal ACWY vaccines were rolled out initially by states, and then the Commonwealth said, okay, they look as though they work pretty well, even though they'd been licensed for some time, so they were then able to put them on the national program and de-risked it. And then finally, is it effective and is it safe in real-world populations in the long term? And that's really important. I'll show an example of where that was not necessarily the case in terms of loss of efficacy. So this, you need to have pathogen surveillance, so that's our communicable disease network of Australia, for instance. Disease-based surveillance, so looking at hospital admissions or GP presentations, depending what the disease is, or in COVID, just anyone who tests positive on a rat, and safety surveillance. And these things change with time, and clearly now we have not great data on community transmission and infection with COVID, because people are doing rats but not notifying it, or not even bothering to do a rats because then you can't go to work or get on a plane or whatever. So we need to think about all these things when we're trying to develop a vaccine. So why are some vaccines not successful? And I'll give RSV, I think, is a good case study here, but we could have actually talked about tuberculosis and BCG. And in fact, this journey started almost 60 years ago in the NIH. These are the guys who discovered the respiratory syncytial virus, and that was just at the time when polio vaccines had been developed. And you remember there's two polio vaccines. One's a live attenuated virus that infects the gut and causes mucosal immunity and systemic immunity, and the other was an inactivated vaccine made by Jonas Salk in the US. And what they did is they had just discovered the RSV virus. They were able to isolate it from infants, and so they said, well, we'll do what we've just done for polio, worked so well, we'll inactivate it with formalin and then we'll inject it and we'll protect the babies. And so this was one of the first studies. In these days, the studies were done in children living in an orphanage near the NIH near Washington, and they were one of a number of studies that looked at this. But at that time, they didn't know anything about the structure, and I'll go into that in a bit more detail. And this was the results. So this was from that paper that was published in 1969. It was done in the early 60s, so almost exactly 60 years ago. You can see how many children are at risk for infection and how many get infected. So this is the RSV inactivated lot. This is their control vaccine, para-influenza virus. So they were also looking for developing a para-influenza virus vaccine, and this was their trivalent one. So they had essentially two risk groups at different stages. And you can see here that a quarter of the children, so 20 infants, 5 of them got infected and 4 were hospitalised, whereas in the control group, so these are kids in the same orphanages, so they should get the same exposures, only 2 got infected and none got hospitalised. And the same happened the following season. So overall you see that, in fact, in this one even more of them got infected, so it was but still had this very high rates of hospitalisation, and much more so than the control vaccines. So 65% and 80% of those who were infected got hospitalised, compared to 60% and 8%. So it didn't work. And not only it didn't work, the children were sicker if they had had the vaccine. And in fact, there were several deaths in this study. So quite tragic. So this is an example of vaccine-enhanced disease. And this actually pointed to what one of the issues were. This was actually in a different study, so in California this time, and this one they actually looked at the autopsies of some of the children who had died, and what they showed was that, including some of the children that had died, and they could see here that they had more severe disease. They both, if you're hospitalised, they both got X-rays, but they had a much higher degree of eosinophilia, and they clearly had more virus isolated than they did in their controls, who were hospitalised with RSV as well. So these were not from the actual trial, but from kids who were hospitalised at the same time. So, and this actually suggested that they got a biased Th2 response that actually enhanced their disease. So a bit like in asthma. So in asthma you get increased number. You can get in young children, particularly atopic children, increased numbers of eosinophils, and so this was driving a Th2 response. And they didn't understand why. Well, but still, why didn't it protect? And I'll show you why that was the case. And so there was no studies after that for about 15 years. 25 years. So, and then they started to make, instead of going with whole inactivated viruses, they did subunit vaccines, so using the fusion protein or combining several proteins within the vaccine. They tried live attenuated vaccines, so these ones that only grow in the cold, so they don't grow at body temperature in the lungs, so you get replication in the nose but not in the lungs. And there's an influenza vaccine, flu mist, that's used in the Northern Hemisphere that is very successful and was highly effective. And then they started to look at taking particular genes out of the virus to see if they could enhance the immune response, or looking at a different type of using multiple peptides, or using a vector with in this case, para-influenza with the RSV. None of these vaccines made it to phase 3 trials. They all failed to elicit high degrees of neutralising activity. So it wasn't just the formalin inactivation that was a problem with those original studies, because these didn't have the same inactivation. So why couldn't they make a vaccine that worked? And in fact, it wasn't really solved until about 10 years ago, when James McClellan, working with Barney Graham in the NIH, actually was able to crystallise the protein, so isolate the fusion protein. And what they noticed when they did that was that there was 2 distinct structures when they purified it from an RSV virus. And what they found was that this one was only present in a pre-fusion. So this was before the virus had infected a cell, okay? And then this was the form of the protein. So it completely changed the protein structure when it had bound to a cell, to allow essentially the virus to punch a hole in the cell, inject its RNA, and then replicate and cause infection. So this had not been known till now. So what they hypothesised would actually all the previous vaccines, because this is actually I won't go into too much detail, but this is the low energy. So this is like a spring that sort of snaps as soon as the virus infects the cell. So it's held stable in the virus and it infects it and punches a hole in it and enables the virus to infect. This is a schematic that was in a recent editorial in the New England Journal showing that this is in pre-fusion, so this is where you bind, so this is the pre-fusion protein binding to the cellular receptors and then altering its structure and then punching a hole through the host cell membrane. And this is showing how it's a trimer, so there's 3 strands of proteins in here. And so what Barney Graham was able to do was he able to stabilise it in this structure. So by putting in these stabilising mutations, it couldn't unfold itself and take the post-fusion state. And so this was a way of maintaining the protein in its pre-fusion state. And in fact, that led to the development of a number of different vaccines. The original Novavax vaccine that was thought not to work because it was still a pre-fusion, and then 3 confirmed pre-fusion structures, one with an adjuvant, one with both the RSV and the RSA and B strains, and then an mRNA vaccine made by Moderna. Studies in very large adults as well as some viral vector vaccines, again using this pre-fusion protein or multiple antigens. And this is the results. The vaccines work incredibly well. So adults with any older adults with any acute respiratory infection, sort of 60 to 70% protection where they look for it. And then this approaching 80% with more severe RSV disease in older adults, and increasingly with with more severe vaccines. So highly effective vaccines for the mRNA, the 2 protein vaccines, one with an adjuvant, and then this adenoviral vector vaccine. Again, very good efficacy suggestion. These have all been published this year, apart from the Moderna one, which will be published soon. So that was how they solved that issue. And obviously, I think I showed you the slide last week, it also works well when you use it in pregnancy to protect young infants. Not only did it do that, but it also enabled them to develop monoclonal antibodies that bound to this pre-fusion construct and select out the ones that didn't bind to the post-fusion. So some, like Palivizumab, actually bind to both. That's Synergis, that's a licensed product. You can see there's epitopes on both the pre and the post-fusion. It works 50-60% prevention. And then we've got Nirsevimab, which is now licensed and has very high levels of efficacy, around 80%. Against half-lives. And you can also make these monoclonals work better by getting them to recycle. So you can change the FC fragment here and actually mean that instead of getting destroyed, it gets recycled. So instead of lasting for a month, as Palivizumab does, or Synergis, it lasts for 5 months. So this is enough to protect babies throughout an RSV season. So this is how structure-based design has informed both vaccines and monoclonal antibodies. And importantly, these principles are exactly applied to spike protein in COVID. So in the SARS-CoV-2 virus, the exactly same thing happens. It's a pre-fusion, the pre-fusion spike protein is the one that is effective in eliciting antibodies. And so everyone made sure they included those stabilising mutations as had occurred for SARS-CoV-2. And there's now 2 monoclonal antibodies that we also imagine will be highly effective. Okay, so that's how you eventually overcome obstacles and design an effective vaccine or monoclonal antibody. So what about ongoing? You've got a vaccine license, you can set and forget. You know, it's licensed, looked at all the data. Well, no, because it's only in 30,000, 40,000. You need to continue to understand where we go with our current vaccines. There might be rare events. Guillain-Barré syndrome, for instance, occurs about 1 in a million after flu vaccines. Interception, 1 in 10,000, 1 in 20,000 after rotavirus vaccines, and febrile convulsions. I'll tell you the story of this with influenza, a particular influenza vaccine in 2010. Also, we now have OSVAC safety that arose out of this particular safety issue that I'll talk about. Importantly, this reflects real life use. So both the variability in the preparation, stability and storage, so do GPs keep it at a cold chain? This was a real problem with the original COVID vaccines. They had to be deep frozen, so minus 20. The general practices weren't ready for that, so they had to develop the data that showed that you could keep it at a fridge temperature. And you're using it much more broadly. So you tend to focus on healthy young infants, older adults, pregnant mums. You tend to exclude the people that you think might give safety signals. So you really want to see, is it safe and does it work in those groups? And that's important, both from a point of view of effectiveness, but also from the point of view of safety. And we can talk about the COVID experience there as well. And what about effectiveness? This is really important. This is one of the challenges, is we know for many of our infections, you get variation. Flu changes every year, okay? Or almost every year we have to change the flu vaccine. SARS-CoV-2, we're changing it every 6 months. We have a number of different national networks that look at, this one's for meningococcal disease, this is invasive pneumococcal disease, this is our general notifiable disease. You can get really good up-to-date notifications. You need to have GP surveillance for flu, there's a number of those, and rotavirus. And we're involved in hospital networks looking at pertussin infants, varicella, influenza, RSV, meningococcal disease. So there's a lot of ongoing research to do these things, and I'll give you some examples. So this is what happens when vaccine safety goes wrong. So this was in 2010. So Fluvax, made by CSL, was a vaccine that, as you can see here in this article, this is Saba Button, who was previously a healthy girl, got her influenza vaccine, ended up with an encephalopathy, seizures, and is now severely disabled. And her mum, understandably, was felt it was her duty to tell people her story, so the dangers of getting flu vaccines. And this was a sort of the take in the media at the time. So why were we giving flu vaccine to lots of people, particularly those in WA? Well, you can see here, so this is, that first thing, is this a disease, which are the most common age groups? So you can see here that in Australia, you actually get less flu in older people than you did in under 5s. Okay, so there's much more hospitalisations in, much more notifications in younger people than there is in older people, although hospitalisations are pretty similar with the very old elderly. But remember, we give everyone over 65 the free flu vaccine. So this was one of the reasons, and if you looked who was at highest risk, it was the children under 2. And in WA in 2007, we had these 3 young kids at the start of the flu season who died of influenza. They were previously healthy preschool children, and so that led WA to roll out a flu vaccine and provide free influenza vaccine for all children from 2008 onwards. And in fact, it worked very well and better than the elderly. So this is work by a colleague of mine who worked with me on looking at effectiveness across a vast range of children, a number of years, children with comorbidities, preterm children, Aboriginal children, and very young children. You can see here the very youngest children actually had the highest efficacy, but overall better efficacy than was seen in the older adults. So this was one of the reasons why this vaccine is now on the National Immunisation Program 10 years after WA first put it on for free for all children. But in 2010, I was actually on holiday at Rottnest, and I got a phone call from a medical friend of mine saying, I've just given my child their flu vaccine, and they had a febrile convulsion that night. They were really hot. Should I give them the next vaccine? I said, look, it was probably another virus. Don't worry too much. If they're well, you should be able to give them their flu vaccine, the second dose, because you need 2 doses when you first have your influenza vaccine the first year. And then after that, you just need a single dose. And then I came back from my holiday, and actually, everyone I talked to in the emergency department said, actually, we've seen quite a few people who've had febrile convulsions after their flu vaccines. And so we actually set up surveillance to say what was going on, and we're able to demonstrate that actually up to 1% of children were receiving flu vaccine were getting febrile convulsions. And they only occurred with this one brand of vaccine, so Fluvax or Fluvax Junior, which was for younger children. you can see here, this was, overall, these are the ones that we were able to associate with the vaccine, and none out of 4,000. So very significant difference. And then when we went and did telephone surveys, so going to mums who had their babies vaccinated at Riola Street Clinic, we were able to show that they were, about half of them were having very high fevers, much more so than the other vaccine brand. And this was in retrospect, so, and you can see a lot of them having rigors as well, so significantly systemically unwell. Whereas other things that you wouldn't relate to it, like headaches, rashes, or local reactions, there was no difference between the groups. And so clearly there was something in this vaccine. Now this is 2010, so the difference was this year that had to include the pandemic swine flu strain. So that was 2009 was the swine flu pandemic, and so they had to include that strain in the new vaccine. So this was the first year, and WA was the first state to do this, although there was similar cases to that encephalopathy occurring in another child in Canberra and a death in a preterm infant in Brisbane. And in fact, we were able to show, as others did, that actually the result was that they hadn't, the splitting process meant that there was more RNA in these vaccines than they had previously. And that was related to particularly the pandemic strain, but possibly also to one of the other strains. So essentially they were causing the flu. So this vaccine is no longer used, and the manufacturing processes are specifically, they check on this how split the viruses are. But of course you can imagine after something like that, the coverage that was occurring, even in children who were at high risk, was much worse for a number of years. And it wasn't until 2018 that you sort of essentially had a new group of parents who hadn't experienced this, that coverage was able to be improved. So this was a real problem, and obviously around Australia it was even worse. We had at least reasonable coverage in these high-risk populations of Aboriginal children and those with chronic neurological heart and lung conditions. So this is what happened. But one thing that did come out of it is that a GP said, look, I was giving all my patients this flu vaccine, but they were presented into the emergency department with their febrile convulsions, and so I didn't find out till a week later when I got the letter that they had been sick. So I was continuing to give the vaccine. And he was an IT-savvy GP, so he got his mate who said, well, we can just send them out an SMS survey like we did to the children in Riola Street. And this has now become a national real-time vaccine safety surveillance. So this year, 215,000 people who've received a flu vaccine have got an SMS to say, did you have any problems? Did you see, if so, yes, what were they? Did you see a doctor? Yes. Did you get hospitalised? And so we can be very certain about how many of them are experiencing one problem, how many are visiting their GP, how many take a day off work. So did any of you guys get the SMS when you had your COVID vaccines? Yeah, yeah, down the back. So you had to tick the box, and it was only in certain clinics. But with COVID vaccines, it's similar. We've been able to demonstrate that the current strains of vaccine that we're giving for boosters are causing less problems. And we can say other things like pregnant women don't complain as much about the vaccines as healthy young nurses or healthcare professionals. Older people tend to report less symptoms than younger people, and infants tend to report a few more symptoms than older children. So it's so this now can be done for any vaccine that's been given in GPs if you're part of this Smart Vac network. So it's a great way of getting safety. So what's the other way? Well, it might be because of waning immunity, and I'll show you the example from the pneumococcal conjugate vaccine. So pneumococcal vaccine, conjugate vaccines were introduced in all children in 2005 and then changed to increase the coverage, particularly because of a 19A strain in 2011. But you can see here in the blue are children having vaccine failures, so getting disease, invasive disease due to the vaccine strains in that first 7 years. Very few, considering 300,000 babies are born each year. So very, very few children. But you can see following the introduction of the 13-valent vaccine, we started to get a significant number of vaccine failures that were occurring predominantly in the first and second year of life. And you can see the numbers here of all the overall vaccines and what age groups they're occurring in. So predominantly they're occurring in the 12 to 36 to 48 months. So not in the first year, but after that. Whereas before there'd been virtually none in these age groups. And these are the overall numbers of vaccines that are occurring in, and this is all. So about half of them were now due to vaccine failure. So we'd lost all those gains that we'd made following the initial introduction here of the 13-valent vaccine. And these tended to be kids with pneumonia and MICR, and particularly 19A and 3, which are the two of the serotypes that were causing these vaccine failures. And at that stage, we weren't giving a booster at 12 months. So we changed our vaccine from 246 to 2412, which is what they've been using in the UK. High-risk infants still get that 246 plus a booster at 12 months. And now these new vaccines are coming along that have completed phase 3 trials. So it's clear we're going to we'll have a lot more strains. It may be less immunogenic, so we'll still need to continue to follow up this surveillance to see if there's any issues. And this also highlights the other issue for many vaccines. How many strains can you cover? And so there's 90 different pneumococcal serotypes, so there's still another 60 to go after you've got these ones covered. So it is going to be very difficult to provide absolute coverage against pneumococcal disease. So what about the future? Well, clearly dealing with SARS-CoV-2 variants causing COVID-19 is a problem. We still have 200 people being in hospital with COVID in WA in any particular week. Not so many deaths as we saw previously, but still a clearly ongoing burden of disease and not really a clear seasonal component. And I think we're starting to get a new surge because I've had 3 people ring in sick today to say they've got COVID and they're not coming into work in my department. They've all just been at a conference in Melbourne, so I don't know whether that might be part of it. We need to use the new technologies that have been developed for RSV and for COVID vaccines to use them in other diseases, and I'll talk a little bit about that. Our understanding of biology is changing completely. Do you guys understand omics? You've heard about genomics, okay? So that's where we sequence everyone's genes, and so we can work out, do you have a gene that creates more susceptibility to a particular disease? So if I have a child who I don't know who's got an immunodeficiency, I can now send off their DNA and get a result back in about 6 to 8 weeks saying they have these genetic defects, or no, we can't find any genetic defect associated with immunodeficiency. But it also applies to the bacteria. So we also now can sequence every virus and bacteria that comes into our lab. And so we're now doing this to help with contact tracing. We know you gave this to this person because it's exactly the same strain. But also our immune system. We can now look at 50, 60 different cytokines that we're producing in response to an infection. Or we can look at proteins. We can look at hundreds of different proteins, all in a very small sample, less than a drop of blood. We can look at our microbiome, so what's associated with disease or health from our microbiome. So we can now start to custom design vaccines and other live bacterial products that actually might be able to prevent infections. And I'll give one example of that. Human challenge models. So you can now challenge healthy adults with an infection and see whether vaccinating them will prevent a disease. So it started with malaria, and there were studies done in Queensland, actually, was one of the first places to do this. But we can now also do it for a number of other conditions. So RSV, pertussis, pneumococcus actually is not in here. Group A streptococcus, I don't know if Alma talked about that. Influenza. So if you think that your drug or your vaccine is going to prevent these infections, you can actually now do it. So we're currently doing a study of a nasal pertussis vaccine where you inject a live attenuated whooping cough up children's noses. Well, they've just shown that you can actually, by using it, you can stop healthy adults getting whooping cough and not getting infected. So clearly this de-risks going to a phase 3 study because you've already shown it works in the human model without having to vaccinate tens or hundreds of thousands of individuals. In fact, there's now COVID challenge studies also occurring. So healthy adults in the UK volunteering, all for the good of man, and you also get paid quite well to be board in a challenge unit for 2 weeks, right? Because you can't let people out if they're carrying these pathogens. So that's making a big difference. So almost every RSV vaccine is now going through these challenge studies. And also you learn a lot about infection. You learn that actually when you first get RSV, you get a lot of neutrophil count if you're going to get infected. and it takes you somewhere between 3 to 4 days to actually start having symptoms, whereas for flu it's only 48 hours. So we then understand a lot more about pathogenesis, so then we can go back and design a better vaccine. However, a challenge, most vaccine preventable deaths still occur in low and middle income countries that are either too poor to afford the vaccine, or even if it's given to them, have struggles to get enough coverage of the vaccines, both for logistic reasons, hard to reach people, people disparate across a large range without easy access, cold chain issues, particularly in tropical countries. Vaccine hesitancy is also an issue, and simply lack of investment in distribution, and certainly very little in terms of surveillance that's reliable. And unfortunately, a lot of our other major vaccine targets that are probably the next stage of vaccines include HIV, TB, malaria, group A strep, dengue. These are really diseases that are most common in low and middle income countries, and we don't really understand much about pathogenesis or the correlates of protection. So this is a real issue. So it's not clear what the model is going forward, and I'll talk about some of the challenges with that. So this is just an example of some of the different design strategies that have been used for developing COVID vaccines. So mRNA vaccines have suddenly come out of nowhere. There's also DNA vaccines. These aren't as good as RNA vaccines, interestingly enough. We were involved in one study where you get very little antibody response after 2 doses of a DNA vaccine. You get a better response and protection after 3 doses, and you get T-cell immunity induced, so you probably don't get severe disease, but it doesn't appear to be very good for protection. So maybe that one should have a cross through it. Nanoparticles and virus-like particles. So HPV vaccine was the first to really, that's why that's so effective, because it self-assembles into a virus-like particle. But they've also done that for SARS-CoV-2. Viral vector vaccines like adenoviruses that are either replicating or non-replicating. So this is a Chadox vaccine and also the Janssen vaccine. Protein and peptide subunit vaccines, so the Clover vaccine that I showed you last couple of weeks ago, but also Novavax that's been used. Live attenuated viruses now becoming more important, particularly for nasal delivery, and then immune therapy, so designing dendritic cells and cytotoxic T cells. They're mostly still, and this is the number that are in development, and actually this is sort of, oh, sorry about that. But you can see here that there's hundreds in clinical trials, up to 200 now, so this is March 2023. 183 in clinical trials, 68 in phase 3 trials, and 11 have been registered. So here again you see this drop-off in the sort of ability to get a vaccine licensed is still very difficult, and it's kept up to date by the WHO. But this is the other big issue. So this is the variant soup. So this is in Australia, looking at the different variants that were causing diseases, and so this is the percentage. So this is 100%, so clearly during the Delta wave, it was all Delta, but that only lasted 3 months, and here comes Omicron. 1, 2, 4 didn't really get a good go, 3 never really did much. Then we got BA5, we got this BA2.75, then we got XBB, so this is now, and this is up to the end of July, which is 84%. We're starting to get some recombinants. You might have heard of Eris as one of the vaccines, and there's now a BA2.86 that's also emerging in overseas. And these vaccines are zipping around the world really quickly. The good news is that these new vaccines, so this is unpublished data in a preprint, but this is an XBB vaccine, so designing a vaccine specifically to cover what's happening at the moment, and this is the vaccine, this XBB 1.5, that's going to be licensed in the next couple of weeks in the United States and will be available in Australia by the end of the year. And you can see this is the original Wuhan strain. So in this study, the red is pre-vaccine, so you've still got pretty good immunity to your original strain that you were vaccinated against, so the Wuhan strain. You've got slightly less titers, so about 1/6 of the titers if you against the BA5 strain, and even less, so what's that? That's an 80th of that against the XBB strain and against all these other strains. And this one's the error strain, and then this is a new, the later strain that they're looking at. But by vaccinating with them with an XBB vaccine, so this is monovalent, okay, so 1.5, you actually get a better response, only about a twofold, but very high levels, higher titers against BA5, and about tenfold increases against all these newer variants, including the Eris and the latest FL1.5, so these recombinant strains. So they have drifted, but you still look as though you'll get as good protection. So potentially you can keep chasing the SARS-CoV-2 virus, improving your vaccines, but maybe we need to look at different. And this is where some of the new strategies, including trying to get mucosal immunity with intranasal vaccines, may be considered. Even intranasal mRNA vaccines are now in trials. So omics, so genomics, so by knowing so traditionally, bacterial vaccines, you went to find what was on the surface, developed an antibody, labelled it with gold, saw if it bound on the surface, said, yep, this one, factor H binding protein, is on the surface, therefore I'll design a vaccine from it. But another way, actually, without doing any of this, you could do this by just knowing the genome. You can then get all your candidates that you can, by examining the structure, know that they've got a motif that will anchor them into the membrane, so they're potential vaccines. You can then express it in E. coli, identify which ones look as though they are novel, and then by immunising animals, you can determine which ones actually which ones generate antibodies that kill the organism, and then you can come out with the 4 antigens that are currently contained in Bexsero, that's licensed for infants and adolescents. And interestingly, one of them that had been identified by Old School was also included in this new. So this is called reverse vaccinology. And Rino Rapoli is the guy who was at Novartis, now then GSK, who did this. And it's been used now for a number of other vaccines that are in late-stage clinical trials. But this is a problem. So this is, you can see here that really it wasn't until the 2000s that children in developing countries, low and middle income countries, were getting vaccines that we were getting as routine 20 years earlier. So hepatitis B, this is when they got wholesale DTP, polio, measles, and BCG for preventing TB. Gavi, which is a global vaccine initiative, then came along, and then they started to say, well, actually, we could get developing country manufacturers to make pneumococcal vaccines, or agree that we could, the Pfizers and the GSKs of the world could actually produce the vaccines much cheaper and then give them. And so you're starting to get 50% coverage for PCV and rotavirus vaccines. But all these vaccines that have been reserved for emerging, so pandemic, this is a pre-pandemic slide, or for antimicrobial resistance, how are we going to get them into what's already a crowded schedule where they really struggle just to get these basic vaccines done? You can see how hard it is. Hib and Hep B have been included in the DTP vaccine, so that's why that coverage is up at 80%. But these newer vaccines are much harder. And in fact, coverage of these vaccines during the pandemic has actually come down. So this is a real problem of how we get vaccines to those people who need it most. And in fact, anti-vaccine hesitancy is actually just as big, if not greater, in these developing countries. Okay, so just to finish off, hopefully you will agree that vaccines remain one of the most effective health interventions we have. Better understanding disease pathogenesis and structure and function of and how that works will lead to more effective vaccines, as we saw for RSV. Vaccine safety is critical for maintaining community confidence, and this we need to continue to work on. And I think one of the things that's happened during COVID is that because we had mandatory immunisation for everyone in order to go to the workplace, that actually drove an anti-vaccine sentiment. In the United States, it was driven politically. So Trump was obviously, even though he had to say originally you should get the vaccines, he didn't actually close down the anti-vaccine sentiment. So a lot of Trump supporters also believe that SARS-CoV-2 is just a Democrat and partnership with vaccine companies to put microchips in them. Some of those ridiculous things. SARS-CoV-2 highlights the ongoing challenges we have with many microbes with antigen variation in your target pathogens. investing in these new technologies is likely to really give important opportunities. So we're currently doing a study of a vaccine to stop, that will be eventually given to pregnant mums to stop cytomegalovirus infection. So there's never really had effective cytomegalovirus. And so this is a mRNA vaccine that contains multiple different antigens. So we understand with CMV that there's different stages you want to try and both prevent and stop reactivation. So by understanding that, you can use the mRNA technology to deliver lots of different antigens in the one vaccine. And so this will be a very interesting study to see if it works. Improving influenza vaccines, universal flu vaccines. There's studies at Linear that are doing that. I haven't talked too much of this but really vaccines for the extremes of age. So very young children, so neonates. So for RSV, that's why we do maternal and pertussis, maternal immunisation. For older adults, how do we give that immune system kick? That was one of the really surprising things about COVID, was that those initial mRNA vaccines worked so well in the elderly. That was really the first time we'd seen that. We now have seen other vaccines like Shingrix for preventing zoster, so an adjuvanted protein vaccine, and the RSV vaccines working really well in older adults. Immunocompromise remain an issue, so people with cancer, people on immunosuppressants for their autoimmune diseases, increased risk and also worse response. So how do we improve vaccines for those particular? But also, nobody's safe till everyone is safe from pandemics. But I think we need to continue looking at how we can get vaccines to those people who need it most. RSV, 99% of all deaths occur in low and middle income countries. Okay, so hopefully that gives you a sense of what some of the challenges are and what we're currently facing, and think about where you might be going over the next 10 years if you remain in vaccine research or delivery or industry, massive industry. And I think you'll find in Australia we now have mRNA factories popping up, sponsored by Moderna in Victoria, vaccine development, manufacturing occurring in Queensland for new vaccines for things like chlamydia that are being developed by researchers at the Queensland University, and I suspect New South Wales and WA will not be far behind. So I think there's great opportunities going forward. Okay, so sorry, as you know, I get enthusiastic with these things. Do you have any questions that I can answer, or queries or comments? If you disagree with me, which is always okay.
When the RSV vaccine eventually, or when one of them eventually gets licensed, are you expecting it to be just, say, under 5s, over 65?
No, so it'll be first, so it'll be mothers will be licensed. That's already been licensed in the United States. I think between when I gave the last lecture and this one, it's actually been licensed. There are vaccines being developed for under 5s, for kids older than 6 months. But it'll be the older people, so people over 65 will be the first licensed vaccines that will be given. And probably all three of those vaccines, the 2 protein vaccines and the Moderna mRNA vaccine, will be licensed and be given to people. So we'll need to know which one works best, how can we work that out. So I think there's great opportunities for research in those areas. But one of the things about RSV in older adults was that until people did detailed studies, nobody actually knew how many people were being admitted to hospital because of RSV. So that was really critical. So there's really early epidemiology studies where they swabbed everyone who came through their door, actually found RSV in a significant proportion. Not dissimilar to flu, which is pretty amazing considering how much focus we've had on flu in the elderly. Any other questions or comments? No? Anything else about vaccines in particular that I haven't covered today that you're interested in? For me, it's always about what I have to leave out rather than having stuff I can put in. Okay, all right. Well, we might finish it up there, guys. You're probably ready for lunch. I know I am. And I've got a few phone calls I'll have to answer. Well, good luck with your exams. And you've got exams, I think, have you? A little test? Yeah. Good. Okay, all right. Well, hopefully it goes well and you enjoy the rest of your course. Cheers. Oh, I wonder when I walk home with this.